Assalamu alaikum everyone. How are you all doing? I hope you all must be fine and doing good at homes. Well, this is Animshir and I'm here with another biologist topic about Unit 6, which is regarding enzymes. So you have studied about the structure of enzymes, about the general properties of enzymes, and today we are going to learn the next topic, or we are going to study the next topic. But before proceeding towards the lecture, let's just have a look about the moral lesson of the day. So what is the moral lesson of the day? The moral lesson of the day is always speak the truth. You might have been listening, or you might have observed that you people have been advised since your childhoods that do not tell a lie or always speak the truth. Do you ever think what is the reason behind that? Well, it doesn't only make you a good person, or it is not only a good habit. In fact, it also saves you from lots and lots of trouble. If you do not tell a lie, or if you are habitual of speaking the truth, you will be afraid of doing anything wrong, so that you might not have to tell a lie. So this is a very good habit. It keeps a person away from sinning. It keeps a person away from doing wrong things. So oneself must speak the truth in every matter of life, in everyday life, in all the circumstances, which can make him or her a very good person, a very obedient person, or you can say a very honest person, in fact. So that was the moral lesson of the day. Now let's proceed towards the topic of the day, or the lecture of the day. Today we are going to discuss about the mechanism of enzyme action. We have studied about the characteristics of enzymes. We have studied about the general structure of enzyme. We have studied the different factors which affect the rate of enzyme activity, the uses of enzymes, and today we are going to study the mechanism of enzyme action. How does an enzyme works? Well, before that, I will just give a brief introduction about the enzymes. That what are actually enzymes? Enzymes are actually protein bodies which are used as biocatalysts. Biocatalysts means they speed up the chemical reactions within the living organisms, or you can say they speed up the biochemical reactions. So whenever they are speeding up a chemical reaction, what are they doing over there in that reaction? You know that whenever an enzyme speeds up a chemical reaction, or it is used in a chemical reaction, it works on the substrate and it changes it into the products. Now we are going to see over here how an enzyme works on a substrate and converts it into the products. Here is a brief introduction about this mechanism. That when enzyme attaches with the substrate, a temporary enzyme substrate complex is formed like this. This is an enzyme and this is a substrate. When this enzyme will attach with this substrate, both of these will combine to form a temporary enzyme substrate complex. What will happen after that? Enzyme catalyzes the reaction and the substrate is transformed into the product. Now what will happen over here? This enzyme substrate complex, what is it going to be converted into? The enzyme will not change itself. There will be no change in the enzyme. The enzyme will not be consumed, but it will change the product. This product will be changed by the enzyme. It will break down the product substrate. ठीक है, ये जो product है, or what is we say that this substrate, the enzyme will cause a change in this substrate. The substrate came and enzyme attached with the substrate. Enzyme got itself attached with the substrate. After that, what will happen? The enzyme will convert this substrate into products. It will break down the substrate and release the products. Now here you can see the substrate is released as a product, whereas there has been no effect on the enzyme. Enzyme is not consumed up during the reaction. It only changes the substrate into the products. Clear? So enzyme catalyzes the reaction and the substrate is transformed into product. After it, the enzyme substrate complex breaks and the product is released. So this is a general equation for this mechanism, that E stands for enzyme, S stands for substrate. So ES complex means enzyme substrate complex, which is formed as a result of attachment of enzyme with the substrate. Then this ES complex breaks into enzyme and products, which means within this complex the enzyme converted the substrate into products, and then the products are released and enzyme is also released. So this P stands for products and E stands for enzyme. So this is actually a brief introduction to the mechanism of enzyme action. There have been different propositions about the models, or you can say that there have been different models which have been proposed on the mechanism of enzyme action, which we are going to discuss now. What are those models? Basically, there are two major models which explains the mechanism of enzyme action. Firstly, let's just have a look on the first model, which is also known as lock and key model. The term lock and key, it might make you think of a typical lock and a key, but why these terms have been used for this enzyme model? Why this term has been used for the model of enzyme action? Here we can see why. Well, it was proposed by a German chemist Emil Fischer in 1894. He described the mechanism of enzyme action. How did he describe the mechanism of enzyme action with this model? He stated that both enzyme and substrate possess specific shapes that fit exactly into one another. What does that mean? That the shape of the enzyme and the shape of substrate are very specific, and they fit exactly into each other, like a lock and the key. The shape of the lock is specific for a specific key. Only a specific key can open a lock. All the keys cannot open all locks. A lock can only be opened by a specific key. You cannot open the lock of a car door with the lock with the key. You cannot open the lock of a car with the help of a door key. In the same way, there are different keys for different locks. Like there are specific shapes of enzymes and substrates. Like here you can see in this figure. This is an enzyme, and you know that only a part of the enzyme is involved in catalysis, and that catalytic region is called active site. So the region which is involved in catalysis is this active site. This is substrate. Now this substrate will attach to the enzyme. There is this, you can say this is a triangle shape or cube, corner pointed shape substrate. So there is a pointed shaped active site, which shows that only this substrate can fit into this enzyme. Now they have formed an enzyme substrate complex. After the process of catalysis, enzyme releases the products, or you can say enzyme has converted the substrate into products. Now these are the products. Substrate has been converted into products. After that, the products will be released, and enzyme as well. Enzyme will also be released from this complex, and the products as well. And again you can see that the shape of enzyme, or there is no change in the shape of enzyme or in the structure of enzyme, whereas there has been a change in the substrate as it has been converted into products. So it is clear from the fact that this substrate and this active site both have specific shapes like a lock and key, due to which this enzyme has been given the name lock and key model. And this also explains the enzyme specificity. It also explains the enzyme specificity very well, because enzymes are really very specific for the type of substrate. You have studied that in the characteristics of enzymes, that enzymes are very specific for the type of the substrate which or nature of the substrate with which they are reacting or working. So this was the first model of enzyme action. There has been another model as well, which is actually the modification to lock and key model. What is that model? That is induced fit model. What do we call? We call it as induced fit model. Induced, term induced means to provoke something or to force something into, to fit into anything. Like induced fit, forcefully filled. Okay? You can say, you might understand from the term that force, in which something is forced to be fit into anything. Okay, so this model or this term, we can interpret from this term induced fit model that there is something going to be fit forcefully. So what does this model state? Who discovered this model? Actually, this model was a modification of lock and key model. Okay, this model was actually a modification to lock and key model. What type of modification? This modification was done by an American biologist, Daniel Koshland, in 1958. So the induced fit model is actually a modification to lock and key model, which was done by Daniel Koshland in 1958. What did he say? What was his statement about this model? He said that, the representation of this model is that the active site of an enzyme is not a rigid structure, rather it is modified into the required shape to perform its function. This shows that the substrate has a fixed shape, but active site does not have a fixed shape or rigid structure that cannot be changed. It can be molded. It is not a rigid structure, so it can be molded into the shape to perform its function. Here you can see the shape of active site is a bit rectangular basically, and the shape of substrate is like this pointed one. Now when this substrate fits into this enzyme, what will the active site do? Active site will occupy the shape of the substrate. Now it has adopted, or it has been molded into the shape of the substrate, and again the substrate and the enzyme has been attached, and they have formed an enzyme substrate complex, or you can say enzyme substrate complex. And here when the substrate has been converted into product, so this is an enzyme product complex, and after that the products are released and enzyme is also released, so the reaction or catalysis is over. So this is how the induced fit model works, that active site of enzyme is not a rigid structure, rather it is molded or modified into the required shape to perform its function. It changes its shape. Here is another point about the acceptability of this model, that induced fit model is more acceptable than lock and key model of enzyme action. Earlier, when there was only lock and key model, so everybody believed that this is a model which explains the specificity of enzyme action, but along with that there was a problem related to the rigid structure of active site. Now after the proposition of induced fit model, this model is a bit more acceptable than the lock and key model, because it says that the active site is not a rigid structure, which has been definitely observed via different processes. So induced fit model is more acceptable than lock and key model of enzyme action. So that was all about the mechanism of enzyme action. Now we are proceeding further towards the specificity of enzymes, as you already know, the enzymes are very specific for the type of their substrate or the nature of the substrate. We actually know that there are almost 2000 known enzymes, each of which is involved in one specific chemical reaction. There are almost 2000 known enzymes, and each of that enzyme, for instance, it means the 2000 enzymes are involved in at least 2000 chemical reactions. They are also substrate specific. How are enzymes substrate specific? Substrates are the molecules on which enzyme is going to act. So here are few examples which explains this. Like, there are different enzymes. The enzyme protease, which works on peptide bonds in proteins. Okay? The term protease, from protease you can understand that it is about proteins. It works on proteins, on the breakdown of proteins or on the breakdown of peptide bonds present in the proteins. But it will not work on carbohydrates, or it will not work on starch, which is a carbohydrate, and starch is broken down by another enzyme which is amylase. And amylase will not break down proteins, and protease will not break down starch. So they are substrate specific, that protease will only break proteins and amylase will only break starch. Same is the situation for lipase. It is another example of enzymes. Lipase, the lipase enzyme acts on lipids and convert them or digests them into fatty acids and glycerol. So the lipase is not going to act on proteins or on starch, it is only going to act on lipids. Same goes for protease, it is not going to act on starch or lipids, it will only break down proteins. And same is the situation for amylase as well. So we say that enzymes are very substrate specific. But their specificity also depends on the shape of active sites. So how the shape of active sites is helping the enzymes to be specific? Active sites possess specific geometric shapes to which only specific substrates bind. Okay? Which means that the active site has the specific geometric shape. Here you can see there is an example or the figure. This is an enzyme and it is having this kind of active site. To this active site only a substrate, this is a very different geometric shape. The substrate having a shape quite similar to this active site can attach to it. All the substrates cannot attach. So here we can see this is substrate 1, this is substrate 2 and this is substrate 3. Which substrate will attach with this enzyme, with this active site? This one, it has so many cuts, whereas it only had 2 curves, 2 or 3 curves. It has so many curves, but this substrate also has 2 or 3 curves. So I guess, or everybody knows by looking at the substrate, this substrate will fit into this active site. So this explains that enzymes are specific not only for the substrates but also for the shape of their active sites. So that was all about the mechanism of enzyme action. I hope you all have understood it very well, and you have to learn this topic as well properly, and if there are any queries you can ask the teachers at that moment in the class. That's all for today beta. Allah Hafiz.